Hello. Okay, can you guys see the screen? Can you guys see the screen? Right.
Yes, sir.
Okay. Yeah. So we'll get started. So I'm Arpit. I'm from IIC and I'll be your PMRF TA for this course. I think biology. I think there's one more TA who has another session tomorrow. So first of all, here are some important links. So the first thing here is the link to this session. The second thing is the link to the YouTube channel where I'll be uploading all these videos. All the sessions will be recorded and will be uploaded here. And the third thing is the Google Drive link where basically all the PDFs of these material which will be discussed in the live session will be uploaded. Okay, so we'll first do a recap of whatever was taught in the first week of the course. So the first lecture was on what is biology. So basically, after going through a lot of examples and asking a lot of people about their take on biology, the instructor defined biology as the study of living things and how they interact with each other and their environment. So once we have this definition, then basically there's one important word here, which I'm highlighting is basically living things. So how do we define what thing is living and what thing is non-living? Sorry. So for that, one way we can define things that are living is by defining what are the characteristics of things that are living. So, for example, we might in general say, so for example, here the tiger and the tree are things that we know are living, whereas rocks or pebbles are things that we know are not living, right? So one might say that the characteristics of living things is that they show movement. So for example, this tiger can go from one place to the other. Then the second thing is they require some sort of fuel. So for example, plants use photosynthesis, which is they get basically fuel from solar energy. They make their food using solar energy. And animals in turn are basically heterotrophs, which basically eat other animals to get energy. Right. The third thing that can be a defining characteristics of living things is reproduction. So basically all living things reproduce to produce progeny. And the fourth thing is waste production. So basically the fuel that you use to basically, for example, anything that you eat, any food that you eat or any um, air, for example, the oxygen that you intake, produce by virtue of metabolism gives you energy, but at the same time produces waste products such as carbon dioxide or let's say, uh, for example, birds produce uric acid and all which they excrete out and things like that. However, it's important to note that all these characteristics are not important for something to be living. For example, the tree example here. Tree is something that does not move from place to place. However, it is a living thing. So these are basically loosely all the characters that are uh, shared by living organisms, but not all of them are necessary for life. So next, the instructor talked about how this course is structured. So the first lecture was mostly focused on what are living organisms, what do we mean when we say things are alive, and so on. And the next lectures are focused on the process of science, then talking about the building blocks of biology, the basics of cell biology, molecular biology, genetics, and then we move on to things like evolution, ecology, ecological interaction, public health, environmental issues, and in the end, we finish it off with biology, society, and the environment and how they correlate with each other. So, does anyone have any questions or doubts from whatever was taught in lecture one. Please feel free to unmute yourselves or basically write if you have any doubts in the chat box. Okay, then we move on to the second lecture where we talked about the pillars of biology. So as we talked, there are like two major concepts in biology. The first thing is genetics, which basically deals with genes. So genes are these individual elements that are present inside the nucleus of different cells, which carry uh, genetic material, which basically defines how a cell is programmed or what a cell is meant to do, and so on. And basically, propagation of this genetic material is how we basically inherit characters from our parents, and like basically how individuals pass on their genes and basically, mm, yeah, pass on their genes and fingerprint blueprints of development to other progeny. And the second important concept in biology is something called evolution. So here we see that uh, there's a famous example of Charles Darwin and his finches. So when he went to this Galapagos Island, he found a lot of finches were basically uh, that basically come from the same uh, initial population, but now have diverged into multiple different populations and species, which basically are focused on eating a certain kind of fruit or nut or something. Right. So the process of how this happens, how a single species basically divides into two, or how new species are created, and what happens is basically evolution. So we looked at all of these in more detail. So for example, evolution is more properly defined as the change in the characteristics of a species over several generations. So a species can be a population of single species living in a given area, or the whole species, which is all the populations combined, how they are changing over several generations. Right. Genetics basically, as I said, talks about genes, genetic variation, and how genes are inherited, and how basically characters or traits are inherited by offspring from parents. Next, we talked about the tree of life, where basically the tree of life is basically a depiction of how things have evolved to be where they are. So if you see the image on the left hand side here, so this thing is something called the Luca, which we'll talk about. So the Luca is basically the last universal common ancestor. So it's basically this thing. So this is the universal common ancestor, which was the original form of life, which basically then uh, not the original form of life as such, but is the um, which was an organism which basically gave rise to all the organisms that we are seeing in the world today. So basically, as we saw before, so the DNA is made up of basically all these different base pairs called EGC, basically adenosine, guanosine, and all those base pairs are basically present in DNA. So a single strand of DNA is composed of a series of these different bases, and basically these bases bind with other bases. So for example, yeah, so these will bind with other bases to form a double helix structure for DNA. So what happens during replication is basically this during DNA replication, when DNA is replicated, is basically the strand separates. So we have one strand here, and basically this strand leads to, so we have complementary strands that are coming up. So one strand which is complementary to this, and another strand which is complementary to this, and that's how we basically reproduce. Basically, how DNA is reproduced. So what happens sometimes is during the process of DNA replication, sometimes the bases that are present get modified. So these things are called mutations. So when these mutations accumulate over time, they lead to different characters because like each part of the DNA, so for example, a sequence of ATGC present in the DNA would define how a certain protein is formed or how a certain um, basically this defines what kind of protein is formed from the DNA, right? That basically carries out a certain function. So if there is some mutation in this part of the uh, DNA, what happens is it changes the protein that is produced. So now we have protein 2 that is being produced here, right? And so on. And if that uh, change from protein 1 to protein 2 is something that is advantageous in the given scenario, in the given environment, then that basically individual will right. Can you guys hear me? Sorry, there was a power cut. Yeah. So specifically, yeah, yeah. Basically, these characters are then inherited by their offsprings, and slowly, that's how these some of these offsprings which have better genes or more genes that are more adapted or more suitable for the environment start developing and becoming better and basically overtake the population or the population changes in a way or the other. Right. So this is basically how change accumulates and leads to different species. So what has happened here is basically the last universal common ancestor that we had. Basically, some changes in their DNA structure led to these branching. One branch developed into bacteria, the other one developed into archaea. Then, sometime later, there was another split where basically organisms that some organisms evolved to be multicellular, which are part of this uh, part of tree of life called eukaryota. And basically, what we have is like these three different domains, which is bacteria, archaea, and eukaryota in the tree of life, which was derived from a single, not which basically came from a ancestor which was common to everyone, called the last universal common ancestor. Okay. So when we look at what living things have in common, we can see that living things have cells and genetic material that is common. So for example, all living organisms are made up of these small fundamental units called cell. So which might be different, but basically all of the living organisms will be made up of the same kind of structures. So for example, here we see an animal cell, a plant cell, and a bacterial cell, all of which have slightly different characteristics, but basically they have a certain membrane which separates them from the environment, and there are like regulatory things and there are different organelles and stuff that perform specific functions inside the cell. Right. And the second thing that is common, so even like multicellular organisms are made up of individual cells that come together to form the multicellular organism. And we have unicellular organisms such as bacteria and all, which basically are made up of a single cell. The next thing that is common to all life forms, almost all life forms, is the DNA. Viruses are known to use some RNA, basically RNA strands as the core of their uh, what do you say? So the DNA is basically the set of instructions that tells a cell what to do, what proteins to produce, what all to do, and how to regulate the cell function and all that. So some viruses use RNA for that, but apart from that, most and all life forms across the earth uses DNA. So what this tells us is that basically these four base pairs, guanine, cytosine, thymine, and adenine, are basically the same across different life forms. So all life forms that are present on Earth are composed of uh, a different combination of these four bases. So cell theory is basically what tells us how cells are and what cells are and basically how they are formed and all. So cell theory has three statements. The first thing is all living beings are composed of one or more cells. So we have unicellular organisms which are composed of a single cell, such as amoeba, bacteria, and uh, plasmodium, and so on. And we have multicellular organisms such as humans, uh, birds, and crocodiles, tigers, which are basically made up of more than one cells. The second thing is the cell is the basic unit of structure. So basically, what this means is the cell basically compartmentalizes all the activities or the biochemical things that are happening that we call as life. It compartmentalizes that from the environment. So all the reactions, the metabolism, and everything else that's happening is happening inside the cell and not outside in general. And the third thing is cells arise from pre-existing cells. So cells are the basic units of reproduction. A cell cannot arise from nothing. A cell tends to arise from another cell that was present before it. However, this leads us to think that how did the first cell come about to be? So the origin of life or how the first cell came out to be is basically a major topic of research and discussion with in both like biochemistry and evolutionary biology, and basically a lot of people are still working on it, and it's one of the unanswered problems in science. Right? So we have, like as I said before, we have organisms which can be unicellular or multicellular. Then even in a multicellular organism, every cell of a multicellular organism will have the same DNA. Right? It does not have uh, different DNA for different cells. All the cells of a multicellular organism share the same DNA because they arise from the same cell. So a multicellular organism basically initially was probably a single cellular organism, not a single cellular organism. So, for example, let's say a zygote is how generally humans or other mammals and other sexually reproducing multicellular beings are produced. So, zygote is basically the first cell, which basically divides into two, then four, and so on, till basically we have a fully functioning multicellular organism. So all of them share the same DNA because all of them are arise from the same cell. Okay. However, there are some organisms which can transition from unicellular to multicellular in their life cycle, in their lifetime. So, for example, this amoeba that we see here can have both unicellular growth and multicellular growth, where basically they are, when these cells are individually present, they grow as unicellular organisms. And when there's like basically harsh conditions and all, they come together and basically form a fruiting body and form these spores that can survive during the difficult times. So multicellular organisms have very different kinds of cells that are specialized to carry out different functions. So, for example, we have nerve cells which carry nerve signals. We have reproductive cells which basically help in reproduction. We have different kinds of blood cells. So, these red blood cells carry oxygen, whereas these lymphocytes, monocytes, and all help. White blood cells basically help us fight infection. We have different kinds of muscle cells. Some are voluntary, some are involuntary, and so on. However, the important thing to remember here is all of them share the same DNA. Okay. So just to recap, what living things have in common is They are all composed of cells, and they almost all always have DNA. And these are some certain characteristics of living organisms, which might or might not be present in all of them, but some of them will, of course, be present in every living organism, such as movement, fuel, or basically how they get their energy, reproduction, waste production, and or metabolism. Right. So, as I said before, all life on Earth was derived from this single point of the tree of life called the Luca, or the last universal common ancestor. So what this means is that all life on Earth shares the same four bases that make up their DNA. So even DNA that you find in bacteria and DNA that you find in humans, all of them are made up of these four different uh, bases. So adenine, thymine, guanine, and cytosine. And that means that the genetic information is written for all organisms in a language that is common for everyone. Right. So what this basically means is that it means that we can do things like genetic engineering. So a DNA or a piece of DNA is basically a set of instructions written to basically create a protein or create some kind of product, right? Substrate or product. So what we have done is basically we have produced these hybrid species such as BT cotton or BT brinjal, where basically the BT stands for Bacillus thuringiensis, which is basically a kind of bacteria that basically is resistant to different diseases that are generally affecting the cotton and brinjal plant. So these hybrid plants that we have, the BT cotton and BT brinjal, are resistant to these uh, diseases because of the presence of DNA from this bacteria in them. Right. So you have transferred the DNA from the bacteria into the plants, and now the plants can synthesize compounds which help them become resistant against certain diseases. Right. Then other things such as insulin production and vaccines are basically possible because uh, all genetic information is written in a common language. So, for example, what we do is we generally, for people who are diabetic who need insulin, what we do is we basically use bacteria and we basically give them a part of our genome which helps them produce insulin, and we basically use the bacteria to produce insulin for us. Right. And even vaccines are created in vaccines and antivenoms and so on, antibodies and all are created in similar way. So the other thing is if we basically match up these sequences of different organisms, we can basically understand how we have evolved from them. So for example, if we have the sequence of different animals and let's say different ciliates, different plants, and different bacteria, and so on. And we try and compare all the different parts of the sequence. Then we can understand like which sequence is closer to which one and which is farther from which one. And based on that, we can make a tree like this, right? So, for example, here it shows that these animals and animals and plants probably came much later than these bacteria and archaea, right? Because the number of uh, similarities in their DNA is more compared to the number of similarities between these animals and, for example, let's say this bacteria or this archaea. Right? And it also helps us tie all living beings together because it's a common language that can be understood and written by everyone. Does anyone have any questions from the second lecture? Okay, so we'll move on to the third, where we basically talked about biology and the city. So we talked about urban scapes. So urban scapes are basically a view of the city as an organism and how we as a city connect with nature. So why is this important? So basically, we are seeing a very high rise in the uh, growth of urban population in different places. Even in India, we see that from the 1950s to 2011, there has been like at least a fourfold or a fivefold rise in the total population, and basically there has been a much um, almost kind of similar rise in the urban population and slightly less rise in the rural population. So there is more and more India is going and settling down in urban areas. It has been estimated that by 2050, I think almost 50% of India would be settling in urban spaces. And these urban spaces are generally very small, very tightly knit together, and often have a very high density of human beings. So it's very important to see how we are managing the things that we do in urban areas, for example, like industries and all that are set up and all, how they affect the environment and how what we can do to see if uh, how we can better our impact on the society and so on the environment and so on. So here we see that since 1950 to around 2025, how the urban population has been growing in India. So a bigger dot means more a higher population, a smaller dot means a lower population. So we see that in 1950s there were only a few cities around which had a lot of urban population, and the major one was probably Kolkata. Whereas now in 2000 and especially in 2025, we can see that we have multiple cities which have a very high population density, urban population, and we see that it's also coming up across the country in different areas. For example, in North like Delhi, Kolkata, Chennai, Bangalore, and Mumbai, and so on. And the blue color here basically denotes if that area is a biodiversity hotspot, right? So we see that two biodiversity hotspots are very prominently shaded, which is the Western Ghats and the Sri Lanka, which is also a continuation of the Western Ghats because we see almost a similar kind of life on either of these biodiversity hotspots. So what we see here is basically that when we juxtapose both the urban population density and the biodiversity hotspot, we see that uh, more and more cities are overlapping with these biodiversity hotspots. So uh, more and more people are also going and settling in the cities because of a better life and so on. And this in turn has a lot of effects on these biodiversity hotspots in terms of like the amount of waste that humans generate, the amount of pollution, the amount of basically resources that we use and so on. And other things such as habitat destruction and so on, which basically affect these areas. And also like not everything is just bad because urban places also have a lot of these parks, and some cities, for example, Bangalore and other cities have a lot of lakes and all, which uh, can lead to an interesting combination of wildlife that is also present in the city. Right. So understanding how these urban scapes or basically urban areas with uh, in an ecological context, how they work out is a very important thing. It was probably Karl Marx who first said that, talked of a city as a organism and talked about urban metabolism, which is basically the um, energy exchange between both the like the societal and ecological factors and the economy that is present in a city. So how they interact with each other and how they basically exchange their energy, how we create things from resources, and basically how does pollution affect them and things like that. Basically, the waste we generate and so on. He said that this is akin to basically what is metabolism in a cell, which is basically using up resources to get energy and produce waste. Right. Uh, so basically, since the 1980s or something, this idea has been popular, and many people have been working on this theme of urban metabolism and how we define it and how we basically can better make cities better, right? So that, yeah, uh, yeah, make cities better in terms of like um, using resources and also dumping waste and so on. So when we make a direct comparison between a organism and a city, uh, let's say we have different kinds of categories based on which we can make this comparison. So for example, the first thing is the form. So form is basically how it exists. So a cell or organism has a defined form, and it follows a strict developmental program to come at that form. So bacteria of the same species would look similar to an individual of a bacteria of the same species would look similar to another individual of bacteria of the same species, or basically a human would look similar to another human compared to a human and something else, right? Because humans have a defined form, a bacteria, a single species of bacteria also has a defined form, and that form comes about by using a strict developmental program. A city, however, can be both planned and unplanned. Many cities in India are very unplanned. However, there are different planned cities such as Chandigarh and all, which are also like well planned before the construction of the city. Whereas many cities in India are basically culmination is a culmination of basically people settling nearby and then slowly those areas developing into urban areas. Right. The next category we talk about is input. So inputs in terms of energy. How does an organism get input? So autotrophs basically use sunlight as a source of energy and get their inputs of energy from sunlight. Heterotrophs, on the other hand, basically get their energy by eating other organisms or other animals. A city nowadays generally gets its energy mostly from fossil fuels. So basically petroleum, coal, thermal power, and so on. However, there are also renewable sources of energy that are currently being used. Next, we talk about water. So, a cell or organism uses water in all its natural forms, whereas a city mostly uses water from the groundwater or from a shallow aquifer, let's say a well or a lake, and so on. And also sometimes rainwater and so on. Uh, Then we talk about materials. So a cell or organism is made up of and uses materials that are available near it and that are generally biodegradable because, for example, the food and all that we eat and the food and all that is eaten by different bacteria and all is basically biodegradable because they are using it up. A city, however, is made up of materials such as steel, wood, plastic, and concrete. Right. Uh, when we talk about the outputs that are produced by cells or cities, when we talk about the waste, for example, we see that cells or organisms generally recycle the waste because, like, uh, waste for one organism might be uh, nutrient for the other. So output for one organism or one species might be the input for another species or another organism. In that way, all the waste that is produced by living organisms is generally recycled and used up by other organisms in the environment. City, however, has a lot of waste that is generated and not directly used up. So, for example, sewage and like garbage such as plastic and all. And also, since urban areas have a lot of construction activity going on, the debris, construction debris that is also found or produced because of that. And when we talk about the next category, which is regulation, so a cell or an organism basically regulates its uh, state. So, for example, a cell will always try to maintain itself at a certain pH, or we as humans would always like to maintain our body at a certain temperature, and so on. So that process is called homeostasis, by which organism or a cell tries to maintain its state. Right. Uh, in terms of a city, when we talk about regulation, so who regulates the growth, who regulates how the city functions, and so on, we think that the government is the body that is responsible for doing all that. So we see that there are some things that are similar, some things that are different when we compare an organism to a city. Right. And people have talked about how urban metabolism happens. So, for example, here we see this is basically example of a linear metabolism in a city. So, what's happening is the city is basically getting food, energy, and water from different sources, and it's producing organic waste such as sewage, inorganic waste such as plastic and all. Uh, and basically all these different gases that are being released. Mostly due to us breathing and also vehicles emissions and basically burning of fossil fuels and so on. So what's happening in this linear metabolism thing is basically all these inputs are coming in, they are being used up to power the city, and basically the outputs that are produced are basically in a way toxic or harmful to the environment, but they are just released to the environment. What we want in turn in a city is to have a cyclic metabolism. So where we are basically reusing or using part of the waste that is being generated by the city. So here what we have is food, energy, and water. And again, basically some of the organic waste that is produced by the city, we are basically recycling it. So for example, let's say using sewage as manure or basically uh, trying to make it into biofuel, converting into biofuel or methane or things like that. Then using reusing it or recycling inorganic waste, for example, recycling plastic, recycling metal things that are basically not being used anymore, uh, and so on. And in this way, what we do is basically we reduce the output that is coming out of the city. Right. So next from urban scapes, we basically move into things, move into the study of basically urban ecosystems. So this is the idea of basically applying ecology to the uh, ecology and ecosystem science to basically urban areas. So what we see here is that urban ecosystems are composed of basically uh, three things. So the first thing is the abiotic and the biotic components that are present in the urban ecosystem. For example, the buildings, the parks, the animals, us humans, different pets, and so on. The second thing is the energy flow. How does the urban ecosystem get its energy? For example, we get energy from a thermal power plant in terms of electricity, or we get energy from solar power plant, solar cells, and so on. And the third thing is nutrient flow, where basically how the nutrients are being cycled in a city. For example, how is carbon cycled in a city? Right. So we are basically using up carbon in the form of fossil fuels and so on. Then we are basically mm, emitting it out when we are burning it to generate power or to generate basically when we are using it as fuel for our vehicles and all. We are basically burning it and releasing a lot of carbon dioxide and all into the atmosphere. So how does that nutrient cycling thing work? And not just for carbon, but for other nutrients also. So by defining all these things and by studying all these things in an urban setting, we are basically studying the ecosystem of an urban area or the urban ecosystem. So for example, in this case study, so talked about in the class, we are basically seeing if we can analyze Surat as an urban ecosystem. So Surat falls on the banks of the river Tapi uh, in Gujarat. Basically, what we see here is a lot of these different buildings and factory is on the other side of the river, which is basically Surat. So it's urban ecosystem which has a river going through it, which has a lot of these plants and all, and also a lot of these skyscrapers and multiple buildings. Right. So to characterize the different components of Surat, what we do is we basically take measurements and so on. So for example, in terms of abiotic factors, what we can characterize are things like what is the range of temperature. For example, in Surat, the range of temperature is 22 to 33 degrees, and there are at least like more than 300 days where there is sunlight. Uh, then the second thing would be the rainfall. So the yearly average rainfall is around 1200 millimeters, and the average humidity is around 60%. Then another thing we can talk about in our elevation and how the landscape is. Flat landscape with 13 meters. So that means it's very close to sea level. And this can be seen from the fact that since I think 2014 or something. Second. Yeah. Since 2014 or something, there have been around three major floods in the city. Then, when we are characterizing the biotic component of the city, what we talk about is the human population, where basically we have 4.46 million people according to the 2011 census living in the city. Then within the city, we see that the fauna that is present is typical of urban areas such as like pigeons, crows, and so on. The river and estuary may harbor many aquatic and amphibian species such as frogs and all. Next, we talk about the energy flow, and we see that there are both terrestrial and aquatic food webs because there's a river that's going in the nearby area. And we see that human activity drives both of these food webs, with basically garbage and sewage being the main contributors or the main sources of nutrients for these. So basically, sewage water that is let into the river basically leads to higher amount of nutrients that are present in the river, which basically feed into the aquatic food webs, and also the garbage that we throw away or are basically taken up by these terrestrial organisms such as rats, eagles, and so on, which basically feed on our garbage, and that basically contributes to the terrestrial food webs. Then we have nutrient cycles such as carbon, where basically due to human activities, mainly due to fossil fuel emission and so on, we have perturbations that are caused by these in the carbon cycling. And there's a lot more carbon emitted that it is being recycled. Then we have the socioeconomic context, which basically tells us that why Surat is a urban area and basically how is it growing? What are the reasons because of which it is growing as an urban area? So, for example, Surat is a big hub for processing diamonds, and it has a lot of industries related to textile business and so on. It's also part of the Vapi Ankleshwar industrial belt, and there's a port nearby, and therefore there's a lot of business that's going on in the city, and therefore many people are coming here for these businesses to take part in these businesses or basically supply raw material, use these processed material, and so on. So the city population is basically growing and will grow in the considerably in the near future because of all these reasons. Right. So basically, by looking at all these different aspects of the city, we basically can study the city and basically see how what kind of impact it has on the ecosystem, how it is different from a natural ecosystem that is present in that given area, and what we can do to basically better it. Now, people have also told that because basically uh, there's another way to look at cities. So basically, what's happening is as cities are growing, So, for example, this is NCT Delhi, where basically the red area that you see is all the built up area, or basically the buildings, skyscrapers, and so on. And basically, these colored areas are basically the cropland areas. Then we have vegetation and bare land that is present. And there's some water bodies and fallow land. So what's happening is in 2001, there was a lot less amount of built up land in this area. Whereas now, like in 2017, now it would be much higher also. In 2017, there has been an increase in the proportion of built up land that is present in this area. And that is because the city has been developing. And because of that, more and more people are basically building houses, building skyscrapers for their offices, for their homes, and so on. So basically, what is happening is the city is growing, and with it, the proportion of area that is under built up land is also growing. So what people see this as is basically they think that the city is basically a cancer because it's hello. Do you have any doubt? Hello. Okay. So basically, what people are saying is that the city is a cancer because it's growing and it's basically increasing the amount of built up area that is present in these different areas, whereas it's eating up more into the natural vegetation, the bare lands, and other kind of lands, croplands, and so on. It's are getting destroyed because of this, because of the growth of the city, which is basically negatively affecting a lot of different things. Does anyone have any questions? We are done with the third lecture. Does anyone have any questions? Okay. Okay. So next in lecture 4, we talked about the process of science, right? So first, what is science? So is science just a body of knowledge or is it a collection of just facts? What is it? So the way we broadly define science is a particular way for understanding the world and how it works. So, but then one might ask that what about religion? Isn't religion a way to understand the world and how it works? Right. So, yeah, it's also a way to understand the world and how it works, but they are slightly different. So, before we move on towards the difference, we will talk about a systematic definition of science, like a more organized definition of science. So, which is also the answer to this question. So science is the organized, systematic enterprise that gathers knowledge into testable laws and principles. This is what one of the famous evolutionary biologist Wilson had to say about science. So we'll move and see what's the difference between science and non-science. So as mentioned before, scientific statements or hypothesis are basically uh, should have two things. One is they should be testable, and the second thing is they should be falsifiable. So testable means basically any scientific statement must be potentially testable. So for example, a positive example of this statement that is testable would be that green spaces improve urban air quality. So how can we test this? We can basically go to urban areas which have green spaces and go to urban areas which don't have green spaces and basically measure the air quality, the particulate matter that is present in the air, and so on, other features of the air that define the air quality in those different areas and see if it's different or not. So we have a test to basically test this statement, right? A negative example of a testable statement would be uh, people who have divine powers don't get COVID, right? So this statement is non-testable because we don't have any test to see if someone has divine powers or not, right? How can you say if someone has divine powers or not, right? So that's why it's a negative example. The second uh, condition that all scientific statements or hypotheses must have is called falsifiability, or the statements should be falsifiable. So that means that there must be some observation that could prove our theory wrong or that could falsify our theory. So falsifiable basically here means proves it wrong. Right. So a falsifiable statement must always have a possible negative answer. So the answer cannot always be positive. There must be a negative answer to the question or to the statement. Right. So a positive example of a falsifiable statement would be that the earth is 800 years old. So one thing we can do to test if the earth is 800 years old or not is something called carbon dating, where we basically take different things and we basically age them. We see what is the age of them, and we can do that for fossils and easily figure out that the earth is not 800 years old. Right. And there's also a lot of different other proofs that we can get to disprove this statement. Right. And different other tests that we can do to disprove this statement. The negative example of a falsifiable statement would be I have a guardian angel that looks out for me. Right. So it's not that you can't have a test for this statement. It's just that even if you have a test for this statement, you can explain the outcome in a way that does not disprove your theory. So, for example, let's say that we are trying to test the second statement. What we do is we basically say that if we have a guardian angel, they'll help us get a job. So let's say we have a job interview coming up, and we are basically like, we'll pray to the guardian angel and they'll help us get a job. Now, if we get the job, we are happy because we feel that our idea is positively reinforced that basically our guardian angel exists. Right. That's why we got a job because of them. Right. Now, the second thing that can happen is you don't get a job. Right. So first thing is get a job. Second thing is don't get a job. So in the first case, you are like, yes, happy, your guardian angel exists, right? In the second case, one might be like, okay, my guardian angel is not does not exist or so on. But then again, one might argue that maybe my guardian angel exists and they know that the job that I was applying for is not something that is right for me. So that's why I didn't get it. So basically, now you have another way to say, or another twisted way of saying that your statement is true, even if the result of your test is negative. So basically, even if you have a test, all the outcomes of the test lead to all positive affirmations of your statement, and you can't disprove this statement by having any kind of proper test. So this is called a non-falsifiable statement, right? Then we talked about what are the features of science. So the first thing is repeatability. So anyone should be able to repeat your experiments or your observations and basically arrive at the same conclusions. The second thing is quantification of measure or measurement. So basically any kind of science that we do, there should be some kind of quantification of things and there should be some measurements that we are basically doing. Mm, mm. Basically, let's say we want to see if there's a difference in height between two species of uh, apes or something, then we can always quantify the height or take a measurement of the height and so on, right? So anything that we are trying to test or something in science should be quantifiable and should be measurable. The third feature of science is peer review. So basically what happens is once you have an idea, once you have done an experiment or something, it should be reviewed by other people and approved by other people. Uh, so that basically gives more strength to what to your observation, to your experiment, and so on. And the fourth thing is abstraction. So it's also a feature of science, but it's not that important or relevant. So, but basically, what this means is basically science grows by abstraction many times. So we can basically abstract concepts to get at larger theories that are governing different things. Right. So this is an example of how the scientific process for unfolds when we are doing science. So the first step is basically making an observation. Then basically, once you make an observation, you look at your system and you basically study all the parameters of your system and ask a question that is relevant to your system. Right. Next, based on the question, you are basically formulating a hypothesis or a possible explanation for why you think some, or a possible guess of what might happen and why you think it's going to happen. Right. Then the fourth thing is basically an experiment or a field study designed to uh, test the hypothesis, basically. And then you basically have measurements or observations from the experiment or field study that are collected and analyzed. Right. So results are then interpreted from the measurements from the observations of the experiment or the field study, and results are interpreted to reach a conclusion that may prove or disprove the initial hypothesis that you had. And after you are done with all this, you basically write it up as a paper or as a poster or as a presentation, and you basically communicate it to others so that others are aware of what you found out. Right. So this is the scientific process. So you make an observation, then you ask a relevant question, you have a hypothesis, then you design an experiment, and you do some measurements, carry out your experiment, get the results, and try to prove or disprove your hypothesis using the results, and then you basically write it out and communicate. So as a case study, we'll look at this example where basically um, Thacker et al. Basically looked at what kind of effects wind farms have on ecosystems and on different trophic levels of the ecosystem. So by checking how these wind turbines are affecting ecosystems in the Western Ghats. So Thacker and colleagues basically investigated the environmental impact of wind farms on the Chalekwadi Plateau in the Satara district of Pune. They basically observed that wind farms seem to influence migration patterns as well as the population of birds, bats, and local terrestrial animals. More specifically, they saw that these wind farms basically negatively affect the populations of predatory birds, because generally these predatory birds are flying high in the sky in search of prey and they basically dive down to eat prey and so on. For example, these kites and all, raptors. So presence of these wind farms and the wind turbines basically affect their flying patterns and areas. And so less individuals of these birds are found in areas which have these wind farms. Right. So that is the observation that they make. So the observation is basically wind farms lead to lower bird populations, right? So they make a hypothesis, which is basically if the so basically this is raptor birds. So if the raptor bird population is decreasing due to wind farms or presence of wind turbines, what should happen is the prey of raptor birds, for example, these small reptiles that are being eaten by them, their population should increase in the presence of a wind farm. So that is the hypothesis that wind farms have a positive impact on population of small reptiles. So what they did was they basically studied this reptile called the fan-throated lizard or the Sarada superba. So it basically has a dewlap like this, right? That's why it's called a fan-throated lizard, because it kind of has a fan on its throat. It's technically called a dewlap. DWLAP. Mm. It's very colorful, and it's generally used for sexual attraction and mate attraction and so on, I think. So what they did was basically, instead of doing the study over a long period of time, where basically they initially measure everything and then go on to measure everything as in the population density of both the prey and predator, and then go on to measure the same after wind farms are installed. What they did is they did something called a space for time substitution, where basically at the same time they look at different places, one which has wind farms, one which does not have wind farms. Right. And they basically quantify all these different things. So how much predators are present, how much predators are attacking, how many prey are present, and so on. So what they found out is that in the presence of wind turbines, the abundance of predatory bird decreases. Right. So it's decreasing. So if there are wind turbines, the number of predatory birds is decreasing. Whereas we see that if there are wind turbines, the number of lizards keep on increasing. Right. So this is basically in line with their hypothesis. And that's why they were able to basically accept the hypothesis and say that, okay, wind farms do have an effect on lizards, and they basically positively affect the lizard populations. Right. So these are the findings. So basically, um, abundance of predatory birds and predation attempts by raptors were almost 4 times lower in sites which had wind turbines than sites which did not. Right. The density of the fan-throated lizard was 3 times higher in sites where wind turbines were present compared to sites where there were no wind turbines. Then the lizards showed a relaxed physiological stress response. So another thing that they did is basically they uh, to more strengthen their case and so on, and basically see if the lizards that they are finding in these different areas with and without the wind turbines, are they more relaxed? So in areas which have wind turbines, are the lizards more relaxed compared to areas which don't have the wind turbines? So what they did is basically they went and they caught a few of these lizards, took their blood, and looked at their like stress hormone levels, cortisol levels, and so on, and basically also tried to go near these lizards and try to see how quickly they are responding to humans. So like how close can I get to the lizard without affecting it? So if I can get much closer to the lizard, that means the lizard is more relaxed. If the lizard is more alert and more like cautious, it will run away, even if I'm present at a farther away distance. Right. So basically they did all these tests and they figured out that lizards showed relaxed physiological stress response and relaxed anti-predator responses in sites with wind turbines. So you can go closer to them in sites where there are wind turbines, and basically they have less stress hormones and other stress compounds present in their blood where there are more wind turbines. However, interestingly, they also found out a few indirect effects that might not be positive for the uh, lizard population. So, so what they found is that these pan-throated lizard from sites which had wind turbines had a lower body condition. That is, they were thinner than those that were present at the sites where there were basically no wind turbines. And they found that the color and the brightness of the patches in their dewlap, which is their throat, fan throat, basically uh, was less colorful and less bright in areas which had turbines. And this they attributed to competition between the individuals. So now, because there are not a lot of predation, there's not a lot of predation in these areas with wind turbines. What is happening is these individuals are competing more with each other. And that's why they are basically not able to have better body conditions. Right. They say that the population and individual level changes in lizards are driven by both the direct and indirect effects of the reduced patient pressure from the top predators, which were the birds. And they conclude by saying that by reducing the impact of predatory birds in that area, wind turbines cause a cascade of changes in the terrestrial prey, which is similar to a predator release. So basically, the wind turbines can be thought of as predators of the apex predators of the system, basically predating on the birds, which are basically disappearing, and that's why the lizard population is increasing. So it's like a cascade of changes that is happening across trophic levels because of the introduction of wind turbines. And further, we see that further they say that since the location of wind farms are mainly determined based on the economic rather than the environmental considerations, they stress that basically the consequences of having a wind farm are greatly underestimated because we generally don't estimate the cost of these wind farms to the ecosystem, to the environment, and to the animals that are present in the environment. Okay. Then in another study, what they did is basically they put up in a university coffee room. What they did is basically they have a coffee machine which basically gives out free coffee, and basically on top of that, they had written, let's say, pay whatever you want or something like that, or like pay for coffee, but there's no one present in that area looking at you. So what they do is in here they basically put a random photo. Either it's like a flower or something, or it's a pair of eyes. And they see that whenever they are putting these eyes, they are getting more amount of money from people. So people are more honest when they feel like someone is watching them, even if there's no one and there's just a plain pair of eyes that is stuck somewhere nearby. And if the eyes are angry and all, they tend to pay more. Right. So this is like alternate weeks, alternate images, they put it up, and they're basically seeing how much money is basically they're getting for each liter of drink milk that is consumed by the people. Right. So experiments like this are small experiments that we can also do in our labs, in our apartments, in places where we are staying and so on. Does anyone have any questions? We are done with lecture 4, which was the process of science. Just to recap, basically, this example I wanted to go over again. So the scientific process is basically, we basically have an observation, then we have a question that we are asking, a hypothesis that we form, then we design an experiment, take some measurements, and we interpret the results to get at a conclusion. So here what they did is they observed that these wind farms are affecting populations of birds. They were like, how does it affect populations of small reptiles? Then they had a hypothesis that they have a positive impact on the population of small reptiles. Then they basically did a test where they tested for the abundance in areas with wind farms and areas without wind farms or wind turbines. And then they basically had these results and they interpreted, and their hypothesis turned out to be correct. Right. And then they basically wrote it up and sent it. Similarly, here also, they have a question that do people pay more if they feel like someone is looking at them? And basically, they designed this experiment, simple experiment where they stick a pair of eyes and basically see how people are paying. And they see that when people are looking at, like, when there are like eyes present in the room, even if there are no people, people tend to pay more. Okay. In the last two lectures, we talked about two prominent but often forgotten scientists from India. Right. So the first one is Dr. Janaki Amman. So she was uh, born in Telisheri, which is now Telisheri in Kerala. in 1897, and she was the 10th child of the EK family. So her father's name was E.K. Krishnan. He was an avid naturalist and a sub-judge in Talasheri. He published two books on birds in North Malabar, and he also maintained a lovely home garden, and probably that's how Janaki Ammal got her interest in science and basically life, birds, and trees, and so on. So this basically sowed the seed of affinity towards natural sciences in Janaki Ammal's mind. But 1890s was a difficult time for women, and so she had to do a lot of things to basically get to where she was. First of all, she had to choose education over matrimony because generally in that age and time, women were not allowed to work. Women were generally not working, uh, generally expected to get married and so on. So basically, she chose education over marriage and basically did the intermediate course in Queen Mary's College in Madras. Then she went on to do her honors in botany in Presidency College and then taught at Women's Christian College. Then she won the prestigious Barber Scholarship and basically flew to America to University of Michigan to complete her master's in botany. So upon arrival on America, she was detained because of immigration approval. However, she was mistaken for an Indian princess because of the saris and all that she was carrying, and then she was let through. Right. After she completed her master's in botany in the University of Michigan, she went on to uh, basically study plant cytology and specialize in breeding interspecific and intergeneric hybrids of plants. Then she came back to India after her master's and taught at Women's Christian College. And yeah, okay. So then she went to Michigan to basically pursue her doctoral studies, and where she did basically um, worked on like chromosome studies in Nicantha silotes, Cyclodes. So this plant basically, under the supervision of Dr. Bradley Moore Davis, she worked on the chromosomes of this plant. She obtained the seeds of this plant from two different races. One was the Indian race called Immaculata, and the other one was the American race called Typica. The seeds were collected from different sources, including Wayanad in India and Tennessee in USA. Then what she did was, during the course of her research, she confirmed that the Nicantra uh, had a haploid chromosome number of 10. So basically, each cell has um, 2n number of chromosomes. So each cell has 20 chromosomes in the organs in the plant. Then there was no cytological differences between the Indian and American varieties. That means they were similar to each other. And then she went on to demonstrate that during meiosis, the chromosomes that are present in the cells conjugate side by side, which is also known as parasynaptic pairing. So here we can see that uh, parasynaptic pairing is basically two chromosomes are present side by side and they're basically combining side by side and then interlocking, whereas telosynaptic pairing is basically chromosomes combining end to end. Right. So earlier there was a huge debate of how chromosomes basically pair during meiosis, and she cleared out that debate and she basically showed that chromosomes basically conjugate side by side by the process of parasynapsis during meiosis. Then in 1931, she was awarded her doctorate. She was the first Indian woman to receive a doctorate in botany in America. Then she returned to India again. She basically was a research fellow in the Department of Botany, Presidency College in Madras in 1932. Then she went on to become the professor of natural science in Maharaja's College of Science in Trivandrum, and then went on to work as a sugarcane cytologist at the sugarcane breeding station in Coimbatore. So when she joined this Imperial Sugarcane Institute in Coimbatore in 1934, at that time the sweetest sugarcane that was present that we were using in India was the Saccharum officinarium variety, which was imported from Southeast Asia from countries like Java. So Janaki identified a native plant variety to crossbreed with Saccharum so that it is better suited to Indian tropical conditions so that we can grow that sugarcane here. So she developed a lot of different hybrids like Saccharum zea, Saccharum imperata, and Saccharum sorghum, and reduced our dependency on Indonesian and Javan imports and promoted India's sugarcane self-sufficiency. So we were able to grow the sugarcane that we are basically using. Her research also helped to establish that the Saccharum spontaneum variety of sugarcane had originated in India. In 1940, she started work at the John Innes Institute in England, where she co-authored this book called Chromosome Atlas of Cultivated Plants with Cyril Darlington. And it was a pioneering piece of work which on breeding and evolution of plants, where basically they recorded the chromosome number of around 10,000 plant species from across the world. Then she joined the Royal Horticultural Society in Wisley, England in 1946 as a cytologist. And she was the first salaried woman employed in the society. And she focused on the botanical uses of colchicine and basically developed a magnolia hybrid. So this flower that you see here is called a magnolia. She developed a hybrid, which was eventually named after her, called Magnolia cobus Janaki Ammal. Then returning to her roots, she came back to India, where basically Nehru was determined to set up the Botanical Foundation for Indian Agriculture, and he requested Janaki Ammal to come back to India in 1950 to be the Director of Agriculture. In 1954, Nehru made her the Director of the Central Biological Laboratory in Lucknow. She also reorganized it into the Biological Survey of India, and it was originally established in the 1890s under the supervision of Britain's Kew Gardens. And after Janaki took over the reins of the Biological Survey of India post independence, she wanted it to be managed and run by Indian scientists and kept for India. Her aim was to make sure that modern systematics research or modern taxonomic research on Indian flora can be revitalized and be free of any colonial influence. So, despite her qualifications, which was a master's and a PhD and so on, the government appointed a European, Hermeneglid Santapo, as the director of BSI. Colonialism was still plaguing the bureaucratic systems in India, and thus the plants that were collecting in India were studied primarily by foreign scientists even after independence, even near independence. Right. So once she joined BSI and all, she tried to reform that and made sure that Indians were basically um, managing and running the Botanical Survey of India. Then uh, another thing happened, which was basically uh, the government was trying to build a dam in the Silent Valley area in Kerala. So basically, the dam would have flooded a lot of the Silent Valley rainforest that is present in that area. So she raised her voice against that and started an environmental movement called Save Silent Valley. She wrote to Darlington that I have made up my mind to take a chromosome survey of the forest trees of Silent Valley, uh, which is about to be made into a lake by letting in the waters of the river Kunti. Finally, the government abandoned the project after years of struggle and protest, and the forest was declared a national park on November 15, 1984. However, Janaki had died earlier that year before this thing happened, around 7th of February 1984. So this is a wonderful book, Chromosome Woman Nomad Scientist, written by Preeti Nair, which is basically a biography of Janaki Ammal uh, and how she lived her life and so on. So yeah, I would suggest you guys to go and read this to get more information on her and see what all difficulties she had to go through to basically do what all she did and be what she is. Does anyone have any questions or doubts? Okay. So at last, we moved to the last lecture where we talked about Dr. Yellapragada Subbarao. So basically, who was he? He was the fourth child of Jagannathan and Venkama, who was born in basically 12th of January 1895 in the Bhimavaram district, Bhimavaram area in West Godavari district of Andhra Pradesh. Uh, so this is what the New York Tribune has to say of him, is that you have never probably heard of Dr. Yellapragada Subbarao, yet because he lived, you may live longer. So if we talk about what all he did, to the end we'll understand why this was said about him. Right. So at the age of 14, he had a dream of becoming a millionaire in Varanasi by selling bananas to pilgrims. So he ran away from his house. However, he came back, and in his early life, he failed his matriculation exam twice, which is 10th standard boards. He failed it twice. His mother had to sell her jewelry to send him to Hindu high school in Madras, and he finally passed the exam there. After that, he wanted to join the Ramakrishna mission, but could not join without her mother permissions, his mother's permission. In 1915, he then enrolled in the Madras Medical College, and his medical school fee was funded by his friends and Kasturi Surya Prakash Rao, whose daughter he eventually ended up marrying in May 10, 1919. And because of his nationalist views and Khadija attire, because this was a time before independence, so yeah, because of his nationalist views and Khadija attires, instead of getting a full MBBS degree, he only got the lesser LMS degree from Madras Medical College. Then, but he secured admission in Harvard School of Tropical Medicine in Dr. Richard Strong's school in Boston. However, there was some unforeseen tragedy that happened in his life. So, tropical sprue, which is an intestinal condition, took the life of both his elder as well as his younger brother. And since Oberoi was dependent on his elder brother's contact for a scholarship to finance his Harvard studies, he was not able to go there. And these tragedies later became a driving force for his future endeavors to fight many diseases. So, since he did not have an MBBS degree, he started his career as a lecturer in anatomy and physiology at the Madras Ayurvedic College, and his goal was to heal the sick, for which he believed he needed foreign education from a good university. Finally, after trying, he got the scholarship and could leave for Harvard 2 years later. He paid part of his fee by doing odd jobs like washing of urinals and bedpans of patients during night hours at the Peter Bent Brigham Hospital. He learned research procedures while working on unsuccessful project involving the laboratory cultivation of E. coli, but he was able to learn a lot of lab techniques during that time. By June 1924, he got the Harvard Diploma in Tropical Medicine. Then he entered Harvard Medical School for a PhD program in biochemistry and joined Dr. Cyrus Hartwell Fiske. There, in Fiske's lab, they developed the Fiske-Subbarao method, which was a rapid colorimetric method for the estimation of phosphate in blood filtrates and total phosphate analysis in blood and tissues. The above mentioned method was called the Fiske-Subbarao method and has ever since been used in the world over in the analysis of clinical and biological material. Right. So why this is important? It's because the amount of phosphorus levels that are present in blood plasma, if we are able to quantify that and see that if it is high or if it is low, we are able to detect if there are some metabolic diseases or if there is a chance of other diseases happening. For example, if the phosphorus level in bone is low, that means there is there probably is some softening of bones and possibility of having rickets. And if there's a very high level of phosphorus present in the blood plasma, that means there's some kidney failure that's probably going on, or there's some metabolic distress such as metabolic acidosis or other metabolic diseases such as cretinism and goiter. Right. So this test is very important to test for all these metabolic conditions. Then his Fiske-Subbarao studies led to the discovery of two new compounds in the muscular tissue, which were involved in muscular contraction. Uh, called phosphocreatine and adenosine triphosphate or ATP. So ATP is basically the molecule which basically gives energy to the cell to do any task. Right. So ironically, Fiske and Subarow did not get the full credit for ATP discovery because independently Carl Lohmann also used the method for phosphate analysis and he announced his discovery of ATP first. Although both of them published it in the same year, 1929. Right. In 1930, he was awarded his PhD in biochemistry, and despite his PhD, he was given the position of a teaching fellow, normally available to graduate students working for their doctorate in Harvard. He continued to work on phosphorus compounds and extracted from spleen, liver, pancreas, and kidney, and isolated around 7 to 8 phosphorus compounds. And George Hitchens, who was the 1988 Nobel Prize winner in medicine and also worked in Fisker's lab with uh, Subbarao, had once said about him that Subbarao had isolated several phosphorus compounds, which were most likely nucleotides associated with RNA synthesis, and they had to be rediscovered by other scientists because he was not allowed to publish that work. Right. So again, in 1935, he had to face a very unusual situation. So Fiske, who was his supervisor, was being considered for a promotion from associate professor to full professor. It was known in uh, generally believed in the Harvard Medical School that Subbarow's share of credit for these discoveries was probably more or at least equal to that of Fiske's. But to save the day for Fiske, Subbarao basically declared that he did not make any significant contributions to the idea, but just provided a pair of hands to realize Fiske's ideas in the laboratory. And because of that, Fiske got the promotion and he was remained like unknown for this, less known for this. Then in 1940, he joined as an associate director of research in Leder Laboratories. There, he finally isolated folic acid from liver in March 1945. Folic acid is used to cure pernicious anemia by promoting the growth of white blood cells in the bone marrow. Uh, so basically, he wanted to make this medicine available in general for everyone. So uh, after that, what he did was he basically tried, since extracting folic acid was very expensive, he tried to find other ways to do it. And after a long time, basically, people were able to synthesize synthetic folic acid, which was much cheaper than the original extracted folic acid that he got. But since folic acid cures pernicious anemia by promoting the growth of white blood cells, the idea to treat leukemia emerged from this. So he thought that if folic acid helps blood cells grow, then a compound that stops it should slow down cell growth. And in looking for a compound like that, he found this compound called aminopterin, which was basically a folic acid antagonist and which brought about remission of WBC in leukemia patients. So Dr. Faber tested this on a child suffering from leukemia and it brought remission of WBC. Then, but this was still a bit toxic. So after looking around for more drugs, he finally yielded a miracle drug called methotrexate, which is kind of similar to that, but it is now used to fight several kinds of cancer and treat several other diseases such as immune deficiency, psoriasis, rheumatoid arthritis, etc. It is also one of the drugs that are still used in chemotherapy around the world. Next, uh, he tried to tackle this disease called filariasis, where basically the lymphatic vessels of the leg tend to be swollen in the patients. And it's a disease that is commonly carried by vectors such as mosquitoes and commonly present in tropical countries. So in 1946, he discovered diethyl carbamazine, which is also called as heterosine, which is recommended by the WHO to treat filariasis, and it is also the most widely used drug for filariasis. Then he started a program to basically find a broad range antibiotic. he engaged the retired plant physiologist Benjamin Duggar in this. And finally, they were able to extract aureomycin from Streptomyces aureofaciens from the fungi, from the bacteria, which is basically the first broad spectrum antibiotic, which was effective against both gram-positive and gram-negative bacteria. Soon after, they discovered or identified tetracycline, which is also a broad-range antibiotic. Uh, on August 8, 1948, he died of a cardiac arrest, and he died a stranger to fame because basically none of these discoveries were ever addressed to him, even if he played a very huge role in all of them. Millions who are alive today because of the drugs he dreamed and grew are yet to hear of him. So just to recap, he is also known as the father of medical medicine. And these are the different discoveries that he made over the years, starting from phosphate analysis, the Fiske-Subbarao method, then phosphocreatine and ATP, several phosphorus compounds from liver, spleen, and so on, folic acid, which is used to treat pernicious anemia, and aminopterin and methotrexate, which are basically used to treat leukemia and other forms of cancer, hetrasen, which is used for filariasis, and automycin and tetracycline, which are basically used as broad range antibiotics. So that's it for this week. Whatever was taught in this week. Does anyone have any questions from the material of this week? If not, we'll proceed to discuss a practice assignment. If anyone has any questions, feel free to drop a question in the chat box or basically unmute yourselves and ask. Uh, so what we'll do here for the practice assignment is that we go over each question. So there are 7 questions. I'll first read the question and all the options, and then you guys can write the answers in the chat or basically unmute yourselves and try to answer the question if you know. Don't worry if it's right or wrong. We'll anyways discuss it and move forward. Okay. So the first question is, which of the following statements is or are true about Luca? So option A, it is the last common ancestor for plants and animals. Option B, Luca is the most recent ancestor for all surviving life on Earth. 3, Luca is the first life that originated on Earth. D, Luca is the last universal common ancestor. E, Luca is my best friend. So which of these options do you think are correct? It can be one or more. I see someone typing. You can also unmute yourself and say. Okay. Someone said A and B. So we'll take a look. So Luca, by definition, is the last universal common ancestor, right? So that means that the option D is correct as such, right? Last universal common ancestor, Luca. The second option that is correct is it is the last common ancestor for plants and animals, right? And B is it is the most recent ancestor for all surviving life on Earth. It Okay, yeah. So it will be B and D, not A, sorry. So the basically we have a tree like this, right? Where we basically have archaea, sorry, bacteria, archaea, and let's say we have plants and animals. So the last common ancestor for plants and animals can be somewhere here, right? So this option A is not correct, right? Option B is Luca is the most recent ancestor for all surviving life on Earth. So B is also not true because it says most recent ancestor for all surviving life on Earth. Right. Luca is not the most recent ancestor for all surviving life on Earth. Luca is the most recent ancestor for all life on Earth, whether or not it is still surviving or not. Because there are many species of bacteria and archaea and all, which are basically extinct. There are many species of plants and animals that are now extinct. For example, the dodo and all. But still, the last universal common ancestor is the most recent ancestor for them also. Right. So option B is also not correct. Luca is the first life that originated on Earth. That is also not correct. Luca is present somewhere here. The first piece of life that originated on Earth is not Luca. Option C is also not correct. The fourth option is Luca is the last universal common ancestor, which is correct because that is by definition Luca. And the fifth option is, of course, wrong. So the correct answer here is D. Okay. The second question is, does anyone have any doubts or questions regarding that? Was it clear? Okay. So the second question is, if DNA sequences between two species are similar, that means A, they have diverged more recently. B, they have diverged very early on. C, they are twins. D, they have a common ancestor. Can anyone answer this? Please feel free to write the answer in the chat. Good job, Isha, for answering the previous question. Even if like doesn't matter if it's right or wrong, just write an answer. Okay, we'll go through all the options. So the question is if DNA sequences between two species are similar. So for example, let's say we have a tree like this. We have A, B, and C. So if what this means is basically, so the option C, it's basically they are twins is false because two species cannot be twins. Right. They have a common ancestor. Uh, yeah, they might have a common ancestor, but if the DNA sequences are similar, does not necessarily mean that they have a common ancestor. It might have come about from different uh, places also. Right. The statements that we are left with is that they have diverged more recently or they have diverged very early on. So now let's say we have this kind of tree where basically X is our common ancestor from which there was some mutation and we got A. Then after some time, there was another mutation because of which we have B and C. So now what this means is B and C will have more similar DNA sequences because they have diverged later on than A, because A would have changed much more on this path, whereas B and C have just changed a little because they have diverged recently. So the answer here will be A and not B. Is that clear? Okay, so the thing is, option D would option D is not right in the sense that, for example, we can have two organisms, A and B, which have similar kind of DNA, but they might have evolved differently or they might have come about from different branches of the evolutionary tree. So, for example, uh, if you think of it, uh, insects and let's say birds, right? They have both of them, sorry, birds and bats, right? Both of them have wings, but they don't have a like recent common ancestor because of their wings. So the wings independently evolved in both birds and bats. Of course, they have a common ancestor, but the similarity in the sequence where basically the wings are getting formed, that part does not tell us that they have a common ancestor. Right, the similarity or dissimilarity in the sequence does not relate to them having a common ancestor. Okay. So the third question is genetic engineering is possible because A, biotechnology in recent days has advanced a lot. B, all living organisms have DNA and one can transfer genes from one species to another. C, there's a lot of funding these days. And D, all living organisms speak the same language. Does anyone want to answer this? Okay, yes. Option D is one correct answer. So we'll go through all the options. So option A, biotechnology in recent days has advanced a lot. That is true, but genetic engineering is not only possible because of that. B is all living organisms have DNA and one can transfer genes from one species to another. That is a major reason why genetic engineering is possible because all of us have DNA and one can transfer the genes from one organism to another. That's why we have BT cotton, BT brinjal, and so on. The third thing is also false. And the fourth thing, all living organisms speak the same language is also correct. So the answer here would be B and D. Both of them are correct. Okay, good job. So question four is cells and cities are different because A, cells have linear pathways where output does not feed back into the input. B, cells are living whereas cities are not. C, cell output often regulates inputs or contributes as raw materials for other processes. And D, cells and cities cannot be directly compared. So can anyone answer? Option B. Okay. So as we talked before, option D, which is cells and cities cannot be directly compared, is false because we made a comparison before. Option B, cells are living whereas cities are not, is also not correct because we talked about how cities can be living organisms, how cities can have metabolism, and so on. We drew a lot of parallels between cells and cities. So basically, in the broad term, we can say that a city is a living organism which has fuel, which has metabolism, which produces waste, and so on. Right. Now, the rest options are A, cities have linear pathways where output does not feed back to the input. This is false because things like recycling and all are things where basically the output is feeding back into the input. And option C, which is cell output often regulates inputs or contributes as raw materials for other processes. So that is true because the output of cell often regulates its inputs and also contributes as raw materials for other different processes. Whereas in cities, that is generally not found. In cities, whatever output we have is generally like, you know, it might be recycled and all, but it does not regulate its inputs and so on. Right. The amount of sewage we produce does not equate to the amount of iron that we use. But for cells, the amount of waste that they produce will equate to will basically affect the amount of nutrients that they take up next time. Okay. Is that clear? So the next question is, which of the following statements are true about the scientific method? The scientific method is a method of acquiring knowledge. The hypothesis is always wrong. Discoveries take time and may later prove to be wrong. It is a process to make sure your hypothesis is always correct. And it is a linear process that cannot be modified or revisited. Does anyone want to answer this? Yeah, you can talk. Someone is unmuting. Okay. So option A, the scientific method is a method of acquiring knowledge. That is true. It's a way of acquiring knowledge and seeing how the world around us works. The hypothesis is always wrong. That is false. Discoveries take time and may later prove to be wrong. That is true, as pointed out by Tamil Selvam. It is a process to make sure your hypothesis is always correct. That is also false. Our hypothesis should be something that is falsifiable, and if needed, based on our data, it should be proved wrong also. And the last option is it is a linear process that cannot be modified or revisited. That is also false. Even after we get a result using the scientific method, we basically go back to it and try to ask more questions and figure out different things in the system. So the answer here is A and C. Now the sixth question is the study by Thakur et al. Investigating effect of wind turbines on the fan-throated lizard population found out that A, wind turbines should not be considered as a green source of power. B, there was a decrease in predatory bird population compared to the same site 10 years ago. C, a reduction in predatory bird population may act as a predator release, allowing the lizards to relax. And D, predator release was supported by a marked decrease in the levels of stress-induced hormones in predatory birds. Which of the following is the correct? Feel free to answer. Okay, so option B is not the correct answer. So we'll go through it. So wind turbine should not be considered a green source of power. That is not at all what they are saying. They are not looking at the wind turbine as a source of power. Option B is there was a decrease in predatory bird population compared to the same site 10 years ago. That is false because they did not carry this study over the time period of 10 years. What they did was a space for time substitution, where they basically, in the same year, took different areas which had windmills and areas which did not have windmills. Right. Option C is a reduction in predatory bird population may act as a predator release, allowing the lizards to relax. That is true because a reduction in the predatory bird population lets the lizards relax and also acts as a predator release, as we talked about before. D. Predator release was supported by marked decrease in the levels of stress-induced hormones in predatory birds. This is false. They did not check the levels of stress-induced hormones in predatory birds at all. They just checked the levels of stress-induced hormones in lizards. And the last question is, which of the following was one of Dr. Janaki Ammal's contribution? A. Investigated the cytogenetics of plant. B. Development of the polio vaccine. C. Discovery of chemotherapy drugs. And D. Published works on biochemistry. It's a quite straightforward question. Does anyone want to answer? Correct. So the answer here is A. She worked on sugar cane and other plants. Was also the head of Botanical Survey of India. And she never worked with the polio vaccine or chemotherapy drugs or in biochemistry. Okay, so the answer here is A. Does so that was the end of today's class. Does anyone have any questions or any doubts regarding the assignment or the course material for week one? Feel free to ask any doubts or questions that you have here. Or you can also ask your doubts in the discussion forum of the course, which is present on NPTEL on the SWAYAM website. From there you can go. Good evening. Good evening. Yeah. Thank you. So the assignment you are required to fill in the website. You don't need to send a scanned copy or something. You just go to the portal, go to the assignment, and you can basically select all the answers and submit it there. You don't need to like write anything or send a scanned copy to anyone for the course. Is that clear? Yeah. So I can you're talking about the course assignment, right? Huh. 1 second, I can just show you. Okay, so can you see my screen? So this is the course page, right? So once you basically go to week one and quiz one, assignment one, the quiz will load, and you can basically select options here which are correct or wrong. And after you select all the answers, you can basically click on submit, and basically that is all. You don't need to like write anything or send a scanned copy to anyone or anything. Is that clear? So basically click on this submit answers button after you're done with all the questions. Hi, good evening. Yeah. So the study material is available again on the website. Mm, uh, Whatever I have prepared here, I'll be sharing all this in this link. So the link that I showed you before. Basically, this Google Drive link is where I'll be uploading everything. So I'll just put the link on the uh, group here also so that you guys can access the folder once I upload things. And as for the material of the course, you can access it on like the SWAM website. Okay, so we'll stop here. We'll continue in the next class. Thank you for attending, and I hope to see you next week. Okay, bye. Good night.